Today, we're going to learn about an important bacterium often present in most infections, Staphylococcus aureus. It's common, sometimes troublesome, and rather versatile when it comes to infections it can cause. Let's talk about its characteristics, virulence, how it spreads, and what you need to keep in mind. Staphylococcus aureus is a gram-positive coccus. They are round cocci and stains purple with the gram stain. Unlike gram-positive cocci, S. aureus typically grows in grape-like clusters rather than chains. When grown on blood agar, it often forms colonies with a characteristic golden hue. This golden colour is a key identifying feature. However, it's important to note that S. aureus can also grow as white colonies on blood agar, so colour alone isn't always definitive. S. aureus can be beta hemolytic, meaning it can break down red blood cells, creating a clear zone around the colonies. However, not all strains exhibit beta hemolysis. To further distinguish S. aureus from other staphylococci, several biochemical tests are performed. It is catalase positive, meaning it produces the enzyme catalase. This is demonstrated by the formation of bubbles when hydrogen peroxide is added to a colony. It is also coagulase positive. This means it produces the enzyme coagulase, which causes plasma to clot. This test is a very important distinguishing factor. Another key characteristic is that aureus is DNase positive. It produces the enzyme DNase, which breaks down DNA. This can be visualized on a DNase agar plate as a clear zone around the bacterial growth. Staphylococcus aureus produces a range of virulence factors, including protein A, hemolysins, and leukocidins, which help it evade the immune system, destroy host cells, and establish persistent infections. These virulence traits, combined with its ability to form biofilms and resist antibiotics, make it a leading cause of both community-acquired and hospital-acquired infections. It can be found on human skin, in the nasal passages, and even on surfaces like doorknobs and shared equipment. It can also contaminate food, leading to food poisoning. Transmission can occur through direct skin-to-skin contact, through respiratory droplets, coughing or sneezing, or by ingesting contaminated food. This is why proper hygiene, especially hand washing, and food safety practices are so important. S. aureus can cause a range of infections, from minor ones like boils and impetigo, to more serious conditions like pneumonia, bloodstream infections, bacteremia, and even toxic shock syndrome. Certain strains, like methicillin-resistant Staphylococcus aureus, or MRSA, are particularly concerning due to their antibiotic resistance. Diagnosis of infections usually involves collecting samples, such as nasal swabs, wound swabs, or blood cultures, depending on the suspected infection site. These samples are then sent to medical scientists, who will use various methods to identify the organism, including the biochemical tests we discussed, and susceptibility testing to determine the correct and effective antibiotics. Treatment of infections depends on the severity and location, as well as whether the strain is antibiotic resistant. While some strains remain susceptible to certain penicillins and cephalosporins, the emergence of MRSA has made treatment more complex, often requiring antibiotics like vancomycin, daptomycin, or linezolid. It is important to properly identify this pathogen and apply the correct treatment. Applying correct treatment helps avoid repeat infections, while also avoiding formation of antibiotic resistance. The information provided in this video is for educational purposes only. Stay informed and click subscribe to support our work.